come back. Let's now look at what we refer to as enzyme-linked receptors. Yeah, just like the word enzyme-linked. Yeah, these receptors have a component of an enzyme. Yeah. Now, in their simple basic structure, assuming this is a cell membrane. Yeah, assuming that is a cell membrane, they also have. If you still remember, we said that a receptor has basically two sides. It has what we call the effector site, which is located within the cell membrane, and then it also has what we refer to as the ligand binding site, which is outside of the cell membrane. So in its simple form, it is something like that, yeah, like this. So in its simple form, it is something like this, yeah, and so it has a part. It has a part that is outside of the cell membrane, and then it has a part that is within the cell membrane. Yeah. Now, that part which is within the cell membrane is endowed with what we call enzymes. Yeah. It is endowed with enzymes. That's why we call it enzyme linked. Yeah. It's endowed with enzymes. And so once you have a receptor, once you have a ligand bind here, then we have a series of reactions that go on to activate these several enzymes. So that's why it is referred to as enzyme linked receptors. So they have what we call a cytosolic enzyme activity as an integral component of their structural function, and so once we have a ligand bind to the extracellular domain, or what we refer to as the ligand binding site, it basically causes or brings about a series of changes that will happen intracellularly or within the cell. And so most of these enzymes are examples of these drugs that follow. These are drugs like insulin, drugs like epidermal growth factor, platelet derived growth factor, and all these are examples of drugs that follow an enzyme. And specifically, like for example insulin, the enzyme in specific is referred to as tyrosine kinase. Yeah, and so what actually happens is that, as you can see in that diagram, we have a component outside, what we refer to as the ligand binding site, and then inside we have a component onto which enzymes are attached, yeah, and these enzymes are referred to as tyrosine kinases, yeah, they are referred to as tyrosine kinases. So once the ligand binds, and in this particular context, tyrosine, yeah, in this particular context, insulin. Once insulin binds, so once the ligand binds on both of them, on the alpha, I think those are called the extracellular units are called alpha subunits, and then the intracellular units are actually called beta subunits. Yeah. So once the ligand binds on the alpha subunits, what actually happens is that they combine, they come together. Yeah, they come together, and then once they come together, this basically activates what we call the tyrosine kinase parts of that dimer, and so each phosphorylates the tyrosine on the tail and on the tail of the other peptide, and as you can see in that diagram. And so once the tyrosine is activated, and there is further activation of enzymes that are located, or proteins, yeah, that are located within the intracellular, and once they are activated, then they go and cause further cellular changes or further cellular response, depending on that. Depending on the type of cell, yeah. So if in this particular context, if it is insulin, then we shall see the effects of the different effects of insulin, as we shall see ahead in the lecture. So that is basically how enzyme-linked receptors basically work. Now, the last part of of our receptors are what we refer to as the intracellular receptors. These are mainly enzymes, yeah? They are mainly enzymes, and many drugs basically target them to produce an effect, yeah? Which brings about altering of, either enzymes production, or depending on, depending on the need of the body, yeah? A small lipophilic molecule basically diffuses through, yeah? So in other words, once the drug or the ligand has to go through the cell membrane, yeah, up to where it will come to bind to the intracellular receptor. Yeah, and of course we have examples of steroids. Yeah, steroids are examples of these drugs. They have to traverse the cell membrane, they enter the cell, up to where they have to find their receptors, which are intracellular. So this is what we are saying, a small lipophilic. Why is it lipophilic? Remember we said lipophilic molecules easily traverse the cell membrane. So it diffuses through the plasma membrane to bind to what we call the intracellular receptors. And of course, they've given us examples as steroid hormones. Now, its binding basically triggers a series of conformational changes within the receptor that leads to, changes within the cell. These changes might be, maybe transcription changes, further changes that need to be, for protein synthesis and all that, yeah? So basically that is how intracellular receptors work. And so these receptors can either be located within the cytoplasm, or it can even be located within the nucleus itself, yeah? And in this particular context, most of these receptors that are located in the nucleus are DNA molecules, yeah? And so once this drug has to effect its changes within the nucleus, yeah, of course so that basically means that the receptor is a DNA molecule, yeah, and so you will have things like maybe protein synthesis as a result of gene transcription, yeah, as a result of gene transcription within the nucleus, yeah, and so basically that is how intracellular receptors work, yeah? So in this lecture, we talked about what we refer to as enzyme-linked receptors, and of course we gave examples of drugs that work through this, like for example insulin, and we also said that there are receptors that are located within the cell membrane, yeah, inside, either within the cytoplasm or on the nucleus. And the expectation is that they will bring about cellular changes. If they are located within the nucleus, and so we shall have cellular changes that are associated with that particular kind of cell. If they are located within the nucleus itself, we shall have things like gene transcription, and in the end result we shall have a series of what we refer to as protein synthesis. So basically, in a nutshell, those are the 4 types of receptors that we have been talking about. We talked about what we refer to as the ligand-gated ion channels. We talked about the G protein coupled receptors. We then talked about what we refer to as the enzyme-linked, receptors. And finally, we concluded it with what we refer to as the intracellular receptors. Now, when we come back, we shall still talk more about receptors, and specifically the several terms that we come across when we refer to receptors.